if you stop right now, wherever you are, and just say wiggle a single finger or maybe take one deliberate step back, that action feels immediate, doesn't it? Almost effortless, simple even.
Mm-hmm.
But the biological mechanics, the sheer complexity needed to make that happen, it involves this incredibly sophisticated operation inside you.
It really does. You've got electrical signals racing, massive chemical shifts, bones moving like levers, all coordinated in, well, milliseconds. It's quite something.
It is astonishing, frankly, the apparent ease with which we move around, just walking or even holding ourselves upright. It hides this amazing system, all derived from the mesoderm, as the NCERT material notes.
Yes, a system that's specialized for converting energy and applying force with incredible precision.
And that intricate specialization is exactly why we're here. We're setting off on a really thorough deep dive today, focusing purely on locomotion and movement and drawing everything from the NCERT material.
Right.
Our aim is to get a solid grasp of the fundamentals. How do muscles actually contract at the molecular level? How do our, 206 bones provide the framework? And importantly, what goes wrong when that beautiful coordination fails?
And to begin, we absolutely have to nail down the definitions. The NCERT material is very clear about the distinction between two key terms, movement and locomotion. They sound similar, but biologically, they're distinct concepts.
Okay, let's tackle that straight away. What's the core difference we need to understand?
Well, movement is the much, much broader idea. It's a fundamental characteristic of living things. Think about the movement of your jaw when you speak, or even just blinking your eyelids.
Simple actions.
Exactly. Or even at a microscopic level, like the streaming of protoplasm inside a single cell, like an amoeba. That's movement too. It just means biological activity or a change in position of a part of the body.
Okay, so movement is general. And locomotion?
Locomotion is specific. It's defined as a voluntary type of movement that results in the entire organism changing its place or location.
Ah, so displacement is key.
Precisely. Walking, running, flying, swimming, those are all examples of locomotion. So the key takeaway, as the NCERT emphasizes, is this. All locomotions are definitely movements, but the reverse isn't true. Not all movements qualify as locomotion.
Right, like my arm moving is movement, but unless it helps me walk somewhere, it's not locomotion itself.
Exactly. Our limbs perform locomotory acts, but they also just adjust our posture, which is movement, not locomotion.
That makes perfect sense. Locomotion is movement with a goal, with displacement. And animals use it for, well, essential things, finding food, shelter, mates.
For, crucially, escaping predators. It's fundamental to survival.
Right. So before we can even get to the big stuff, like flexing a bicep or running, we need to zoom right down to the cellular level, because that's where this ability to, shift and push originates. According to the NCERT material, what are the main types of movement our own cells exhibit? I think there are three primary ones.
Yes, that's right. There are three main types: amoeboid, ciliary, and of course muscular movement. And they really show how movement has been adapted for completely different jobs within the body.
Let's start with amoeboid movement. That sounds like it belongs to single-celled organisms, but you said our cells use it too.
They certainly do, especially certain specialized cells. Think about macrophages, which are like the cleanup crew of our immune system, and also leukocytes, the white blood cells in our blood.
Okay.
This type of movement happens using pseudopodia, that literally means false feet. They're formed by the protoplasm actively streaming or flowing within the cell.
So the cell kind of oozes forward.
In a way, yes. It's a controlled deformation.
Yeah.
And the NCERT material makes a key point here. This relies heavily on cytoskeletal elements, specifically microfilaments.
Ah, the internal protein strands.
Exactly. They act like internal scaffolding that can be quickly assembled and disassembled, allowing the cell to change its shape and move in a particular direction. It's less about a fixed structure moving, more about controlled internal flow.
Okay, that's quite different from the next type then, ciliary movement, which sounds much more organized.
It is very different. Ciliary movement happens in our internal tubular organs, the ones lined with what's called ciliated epithelium. Imagine tiny hair-like structures, cilia, covering the surface.
Right.
And their movement isn't random. It's highly coordinated, often like a wave passing over the surface. The main purpose is usually to move substances along that surface.
What are some crucial roles for this kind of sweeping action in us?
Well, two really important examples are given in the NCERT material. First, defense. In our trachea, our windpipe, these cilia constantly beat upwards. They sweep mucus, trap dust particles, and any foreign stuff away from the lungs. It's like a self-cleaning filter.
Essential for keeping the lungs clear.
Absolutely. The second key role mentioned is in the female reproductive tract. The coordinated beating of cilia there helps to gently move the ovum, the egg cell, along the fallopian tube after ovulation.
Fascinating. And this isn't just in humans.
No, not at all. The NCERT also mentions examples like paramecium, a single-celled organism where cilia are used both for moving food towards its mouth area, the cytopharynx, and for propelling the entire organism through water. And even hydra use tentacles, which involve ciliary action for capturing prey and for locomotion.
Okay, so we have the flowing, adaptable, amoeboid movement, the coordinated sweeping of ciliary movement, and that brings us to the third and perhaps the most powerful type, muscular movement.
Exactly. This is the type we usually think of. It's required for all the large-scale movements moving our limbs, the intricate motions of our jaw when we eat or speak, the dexterity of our tongue.
And this relies on a specific property of muscle tissue.
Yes, its unique ability, contractility, the power to shorten forcefully. But, and this is a crucial point for linking back to locomotion, the muscle can't achieve locomotion on its own.
Right, this is where the integration comes in. It's not just muscle.
Precisely. True locomotion requires the perfectly coordinated activity of three systems working together. The muscular system to provide the actual force.
The engine.
The skeletal system, providing the rigid framework, the levers, the attachment points.
The chest.
And absolutely vital, the neural system, which sends the precise electrical commands to initiate, control, and coordinate the entire process. It's a three-part symphony. And when you look at the muscles themselves, the scale and specialization are just incredible. As we mentioned, muscle tissue comes from the mesodermal layer during development, and it makes up a huge chunk of our body mass. The NCERT states it's about 40 to 50% of the body weight of an adult human.
Wow, nearly half our weight is muscle. That really underscores how central muscle function is. Now, the NCERT material highlights 4 special properties of muscle tissue. Can we quickly go through those?
Certainly. They define what makes muscle tissue unique. First, excitability. This is the muscle's ability to respond to a stimulus, which is usually a nerve impulse.
It can receive the signal.
Exactly. Second, contractility. This is its defining feature, the ability to shorten forcefully when stimulated, generating that pulling force.
The pulling power.
Third, extensibility. This means the muscle can be stretched or extended, even beyond its normal resting length. Think about stretching before exercise.
Okay.
And finally, elasticity. This is the ability to recoil, to snap back to its original resting length after it's been stretched or contracted. So these 4, excitability, contractility, extensibility, and elasticity, make muscle the perfect biological engine.
Got it. Now let's talk classification. Muscles aren't all the same. How does the NCERT categorize them? It seems to be based on location, appearance, and how they're controlled.
That's right. There are 3 main types outlined. First up, we have skeletal muscles. These are the ones most closely associated with the bones of our skeleton, hence the name.
The ones we use to move around.
Precisely. If you look at them under a microscope, you see very distinct stripes or bands running across them. These are called striations. So they're also known as striated muscles. And crucially, their activity is under our conscious control via the nervous system. That makes them voluntary muscles.
So deciding to lift something, wave, walk, that skeletal muscle.
Exactly. Their main jobs are locomotion and making changes to our body posture.
Okay, that covers the voluntary ones. What about the muscles working behind the scenes without us thinking about it?
Those would be the visceral muscles, more commonly called smooth muscles. You find these in the inner walls of hollow internal organs. Think of the alimentary canal, stomach, intestines, the urinary bladder, the reproductive tract.
Right.
They lack those cross striations seen in skeletal muscles, so they look smooth under the microscope, hence the name non-striated muscles. And their activity is completely outside our conscious control. They are involuntary muscles.
So what's their main role?
Their primary job is usually transport. For example, they push food through the digestive tract via peristalsis or move urine along. They manage the internal plumbing, essentially.
Makes sense. And the third type, it seems like a bit of a hybrid.
Yes, the cardiac muscles. These are found only in the wall of the heart. Structurally, they're unique. The cells are branched and connect end to end, forming a network.
And their appearance, striated or smooth?
They are striated, like skeletal muscle, but functionally they are entirely involuntary. We don't consciously decide to make our heart beat faster or slower. It has its own intrinsic rhythm, although the nervous system can influence the rate.
Okay, 3 distinct types. Let's really focus now on the skeletal muscle, since it's the powerhouse for locomotion. We need to break down its structure, right from the whole muscle down to the molecules involved.
Absolutely. Let's start big picture. An organized skeletal muscle, like your bicep, is made up of many bundles of muscle cells. These bundles are called fascicles.
Okay.
These fascicles are grouped together and wrapped in a layer of tough connective tissue, collagenous connective tissue called fascia. Now, if you look inside one fascicle, you find the individual muscle cells, which are usually called muscle fibers.
And the muscle fiber itself is a pretty special cell, isn't it? Highly adapted.
It really is. Each muscle fiber is enclosed by its own plasma membrane, but in muscle, it gets a special name, the sarcolemma. Inside the sarcolemma is the cytoplasm, again with a special name, the sarcoplasm.
And I remember reading something about multiple nuclei.
Yes, that's a key feature mentioned in the NCERT. The muscle fiber is a syncytium, meaning it contains many nuclei. This results from the fusion of multiple precursor cells during development. And within the sarcoplasm, the endoplasmic reticulum is highly specialized. It's called the sarcoplasmic reticulum.
And this sarcoplasmic reticulum has a critical role, doesn't it? It's like a storage tank for something specific.
Absolutely critical. It's the main storage site within the muscle fiber for calcium ions, that's Cas2+. The controlled release and then reuptake of these calcium ions is essentially the on-off switch for muscle contraction.
Okay, calcium is key. Now, inside the sarcoplasm, packed within the fiber, are the actual contractile elements.
Yes. Thousands upon thousands of tiny parallel protein threads called myofibrils. These run the length of the muscle fiber. And when we talked about skeletal muscle being striated, those stripes we see, that's actually the visual result of the highly organized repeating pattern of proteins within these myofibrils.
The light and dark bands. Let's define those based on the proteins.
Okay, so you have alternating dark and light bands. The dark band is called the A band, which stands for anisotropic band. This band contains the thicker protein filament, which is myosin.
Band, dark, myosin.
Got it. The light band is the I band, for isotropic band. This band contains the thinner protein filament, actin. And these proteins, actin and myosin, are arranged in a very precise parallel fashion.
And the whole process of contraction is defined by what happens to these bands and their boundaries.
Right. Exactly. The fundamental unit of contraction, the repeating unit along the myofibril, is called the sarcomere. The NCERT defines the sarcomere as the region of a myofibril that lies between two successive Z-lines, sometimes pronounced chrysis lines.
And what is the Z-line itself?
The Z-line is an elastic protein structure that runs down the middle, bisecting each I band. Crucially, the thin actin filaments are directly and firmly attached to these Z-lines at either end of the sarcomere.
Okay. And within the A band, the myosin zone, there are other landmarks too.
Yes. Right in the middle of the A band, holding the thick myosin filaments together, there's a thin fibrous membrane called the M line.
M for middle.
M for middle, exactly. Now think about a muscle at rest. The thin actin filaments extend in from the Z lines and partially overlap the thick myosin filaments at the edges of the A band. But there's a region right in the center of the A band where only the thick myosin filaments are present. There's no overlap from the thin filaments. The central myosin-only zone is called the H zone, named after Henson.
Ah, the H zone. And that's the visual key, isn't it? Its width changes dramatically during contraction.
Absolutely. When the muscle contracts, the actin filaments slide further into the A band, towards the M line. This overlap increases, and consequently the H zone shrinks, or can even disappear entirely in a fully contracted muscle. Seeing the H zone shrink is direct visual evidence of the sliding mechanism.
Okay, structure is clear. Now let's get into the molecular players themselves. These contractile proteins, actin and myosin, are complex structures. Let's start with the thin filament, actin.
Right. The thin filament isn't just actin alone. It's primarily composed of two chains of F-actin-F for filamentous wound around each other in a double helix. Think of it like two beaded necklaces twisted together.
And what are the beads?
Each F-actin chain is actually a polymer made up of many repeating globular units called G-actin G for globular. Now, running alongside these two F-actin chains, in the grooves of the helix, are two filaments of another protein called tropomyosin.
Okay, so F-actin helix plus tropomyosin. What else is part of this thin filament complex?
The crucial regulatory element is a complex protein called troponin. It's attached at regular intervals along the tropomyosin molecules. Troponin is the real gatekeeper. In a resting muscle, one specific part, a subunit of troponin, physically covers or masks the active sites on the G-actin molecules.
Active sites for what?
active sites for the myosin heads to bind to. So in the resting state, troponin and tropomyosin work together to block the interaction between actin and myosin.
Got it. Troponin is the lock. Calcium will be the key. Now for the thick filament, myosin.
Myosin is also a polymer, but it's built from many individual monomeric proteins called meromyosin. Each meromyosin molecule has two distinct parts.
Okay.
There's the heavy meromyosin, or HMM. This part consists of a globular head region and a short arm or neck region. This head and arm structure projects outwards from the main axis of the thick filament and is often called the cross-arm or cross-bridge head.
And the other part?
The other part is the light meromyosin, or LMM, which forms the tail of the meromyosin molecule. The tails of many meromyosin molecules bundle together to form the backbone of the thick filament, while the HMM heads project outwards in a spiral pattern.
And that globular head of the HMM, that's the business end, right?
Absolutely. It has several critical functions packed into it. First, it acts as an active ATPase enzyme. That means it can bind to ATP adenosine triphosphate and hydrolyze it, breaking it down into ADP, adenosine diphosphate, and inorganic phosphate pi. This releases chemical energy.
The energy for contraction.
Exactly. Second, it obviously has the binding site for that ATP molecule itself. And third, crucially, it has the active sites that allow it to bind specifically to those sites we just mentioned on the actin filament. Power, fuel, and the hook, it's all in the myosin head.
Okay, we have all the players and the structure. Let's put it all together now with the mechanism of muscle contraction, the sliding filament theory. What's the fundamental idea here?
The core principle, proposed independently by Andrew Huxley and Rolf Niedergerk and by Hugh Huxley and Gene Hansen, is quite elegant. Muscle contraction doesn't happen because the actin or myosin filaments themselves shorten. Instead, the thin actin filaments slide inwards past the thick myosin filaments.
They slide over each other.
Precisely. The sliding action pulls the Z-lines, which the actin filaments are attached to, closer together, causing the entire sarcomere to shorten. And when millions of sarcomeres shorten in series and in parallel, the whole muscle contracts.
How does this whole complex process get started? It needs a signal from the nervous system.
Yes, it always begins with a signal originating in the central nervous system, the CNS. This signal travels down a motor neuron.
Okay.
That motor neuron makes a connection with the muscle fiber at a specialized junction. This junction is called the neuromuscular junction, or sometimes the motor end plate. The neuron doesn't quite touch the muscle fiber. There's a tiny gap called the synaptic cleft.
And the neuron plus the fibers it controls has a name.
Yes, the motor neuron plus all the muscle fibers it innervates is collectively called a motor unit.
Right. So at that neuromuscular junction, the electrical nerve signal gets converted.
Exactly. When the nerve impulse arrives at the end of the motor neuron, it triggers the release of a chemical messenger, a neurotransmitter, into that synaptic cleft. The specific neurotransmitter here is acetylcholine.
Acetylcholine.
This acetylcholine diffuses across the gap and binds to specific receptors on the sarcolemma, the muscle fiber membrane. This binding event opens ion channels and generates an electrical potential, an action potential in the sarcolemma.
So electrical signal in the nerve becomes a chemical signal across the gap, then back to an electrical signal in the muscle fiber.
Correct. And this muscle action potential doesn't just stay on the surface. It spreads rapidly across the entire sarcolemma and, importantly, travels down in vaginations called T-tubules deep into the muscle fiber's interior.
And what does this electrical wave trigger deep inside?
This is the crucial step. The arrival of the action potential deep within the fiber triggers the sarcoplasmic reticulum to release its stored calcium ions, K2+, into the sarcoplasm, the fluid surrounding the myofibrils.
The calcium floodgates open.
Exactly. Suddenly, the concentration of calcium ions in the sarcoplasm increases dramatically. This is the direct trigger for contraction to begin.
And this flood of calcium interacts with that protein complex we talked about earlier, the one blocking the actin sites.
Precisely. The increased calcium levels in the sarcoplasm allow the calcium ions to bind to a specific subunit on the troponin molecule. This binding causes troponin to change its shape. And because troponin is attached to tropomyosin, this shape change pulls the tropomyosin filament slightly aside, physically uncovering or unmasking the active binding sites on the G-actin molecules.
The binding sites are now exposed.
They are now accessible. The lock has been opened by the key of calcium.
So now the myosin head can finally interact with actin.
Yes. The myosin head, which has already hydrolyzed an ATP molecule and is sort of energized or cocked, it has ADP and inorganic phosphate bound to it at this stage, now has a high affinity for the exposed actin site. It rapidly binds to the actin, forming what's called a cross bridge.
The physical link is made.
Hmm.
And what happens next is the actual pull.
Correct. The binding of the myosin head to actin triggers the release of the ADP and the inorganic phosphate pi from the myosin head. This release causes the myosin head to undergo a conformational change. It pivots or swivels. This movement is called the power stroke.
And it pulls the actin filament.
Yes, like rowing an oar. The myosin head, still attached to the actin, pulls the thin filament inwards towards the M line at the center of the sarcomere. Since the actin filaments are anchored to the Z lines, this action pulls the Z lines closer together, shortening the sarcomere.
And this has those visual consequences we discussed.
Exactly. As the NCERT clearly outlines, during this contraction, the I bands, containing only actin, get shorter, and the H zone, containing only myosin in the center, also shrinks or disappears. Importantly, the A band, defined by the length of the myosin filament, does not change its length. This is key evidence for the sliding filament model that the filaments slide, they don't shorten.
Okay, the power stroke happened. The sarcomere shortened. But the myosin head is still attached. How does it let go and get ready for another pull? This is where the next ATP comes in, right?
This is a really critical point, often misunderstood. After the power stroke, the myosin head is stuck to the actin in a low-energy state. To detach it, a new molecule of ATP must bind to the myosin head.
So ATP binding causes detachment, not the power stroke itself.
Correct. The binding of fresh ATP causes a conformational change that decreases the affinity of the myosin head for actin, and the cross bridge breaks. Then, almost immediately, the ATPase activity of the myosin head hydrolyzes this new ATP back into ADP and pi, re-energizing or cocking the myosin head, getting it ready to bind to another actin site further down the filament, provided calcium is still present and the sites are exposed.
So it's a cycle. Bind, power stroke, detach, requires ATP.
Yeah.
Re-energize using ATP hydrolysis. Repeat.
Exactly. This cross-bridge cycle continues, with the myosin heads acting like ratchets, walking along the actin filaments as long as two conditions are met. Calcium ions are present at a high enough concentration to keep the actin sites unmasked, and there's enough ATP available both to power the stroke and to cause detachment.
And how does the muscle relax then? How does the cycle stop?
Relaxation occurs when the nerve signal from the CNS stops. No more signal means no more acetylcholine release at the neuromuscular junction. The action potential on the sarcolemma ceases. And without that electrical signal, the sarcoplasmic reticulum actively starts pumping the calcium ions back inside itself, out of the sarcoplasm. This requires energy, it uses ATP pumps.
So calcium levels drop in the sarcoplasm.
Right.
Huh.
As the calcium concentration falls, calcium ions detach from troponin. This allows troponin and tropomyosin to shift back to their original positions, once again masking the active binding sites on the actin filaments.
Myosin can no longer bind.
Exactly. Cross-bridge formation stops. The pulling force ceases, and the natural elasticity of the muscle tissue, along with perhaps opposing muscle action, pulls the sarcomeres back to their original resting length. The muscle relaxes.
That is an incredibly elegant yet complex and energy-demanding process. That massive need for ATP brings us to think about muscle metabolism and, inevitably, fatigue.
Indeed. Muscle contraction is one of the most energy-intensive processes in the body. Sustained or repeated activation, especially if it's intense, can outstrip the muscle's ability to produce ATP aerobically, using oxygen.
And that's when fatigue sets in.
Yes. When oxygen supply becomes limiting, muscle fibers increasingly rely on anaerobic glycolysis, breaking down glycogen stores without oxygen. This process is much faster but less efficient, and it produces lactic acid as a byproduct. The accumulation of this lactic acid is considered a major factor contributing to the sensation of muscle fatigue and temporary muscle soreness.
And this difference in metabolic strategy is reflected in the different types of muscle fibers we find, right? The red and white fibers mentioned in NCERT.
Precisely. Muscle fibers aren't all identical metabolically. We have red fibers. They get their color because they contain a high amount of a pigment called myoglobin.
Like hemoglobin in blood, but for muscle.
Exactly. It's an oxygen-storing pigment. Red fibers also have lots and lots of mitochondria, the powerhouses of the cell. They are well supplied with blood vessels. All this means they are geared up for aerobic respiration. They can use stored oxygen and efficiently produce ATP over long periods. They are fatigue resistant.
So endurance activities.
Yes, think posture muscles or the leg muscles of a marathon runner. They are often called slow twitch or aerobic muscles,
Any other type?
the white fibers. These have very little myoglobin, so they appear paler or whitish. They also have fewer mitochondria compared to red fibers. However, they tend to have a much higher amount of sarcoplasmic reticulum and often a larger diameter.
So what's their metabolic strategy?
They rely heavily on anaerobic processes for energy. They can generate ATP very quickly through glycolysis, allowing for rapid, powerful contractions. But because anaerobic metabolism is inefficient and produces lactic acid quickly, these fibers fatigue much faster.
So power and speed, but not endurance.
Exactly. Think sprinters, weightlifters. These are often called fast twitch or anaerobic fibers. Most muscles contain a mixture of both red and white fibers, but the proportions can vary depending on the muscle's function and can even adapt somewhat with training.
Okay, let's shift gears now. We've covered the end in the muscle, but the engine needs a frame, a chassis, to work against. That brings us to the skeleton. Without those 206 bones providing levers and stable attachment points, muscle force wouldn't translate into useful movement or locomotion.
Absolutely. The skeleton provides the essential structural support, it protects vital internal organs, and critically, it forms the system of levers that muscles pull against to create movement. Before we dive into the bones, the NCERT reminds us that both bone and cartilage are specialized connective tissues.
What's the key difference in their makeup?
It's primarily the matrix, the non-cellular material surrounding the cells. In bone, the matrix is very hard and rigid because it's heavily mineralized with calcium salts. In cartilage, the matrix is firmer than other connective tissues, but still somewhat pliable or flexible due to the presence of chondroitin salts.
Okay, now those 206 bones in an adult human skeleton are organized. The NCERT divides them into two main divisions.
Yes, the axial skeleton and the appendicular skeleton. Let's tackle the axial skeleton first. This forms the central axis of the body and consists of 80 bones in total.
80 bones in the core. What does that include?
It includes the skull, the vertebral column, the sternum or breastbone, and the ribs. Let's start at the top with the skull. It's actually quite complex, made up of 22 separate bones.
22. How are they grouped?
They're divided into two main sets. First, you have the cranial bones. There are 8 of these flat bones, and they fuse together to form the protective bony box, the cranium, which encloses and protects the brain.
And the second set?
Those are the facial bones. There are 14 of these, and they form the front part of the skull, the framework for the face, including the jaws.
But aren't there other bones associated with the skull, like in the throat and ears?
Good point. The NCERT mentions these separately. There's the single U-shaped hyoid bone located at the base of the buccal cavity, the mouth-throat area. It's unique because it doesn't directly articulate or form a joint with any other bone.
It just sort of floats.
It's anchored by muscles and ligaments, yes. Then, within the middle ear cavity on each side, you have the 3 tiniest bones in the body, the ear ossicles. These are the malleus hammer, incus anvil, and stapes, stirrup. They transmit sound vibrations.
So 22 skull bones, plus 1 hyoid, plus 6 ossicles, equals 29 bones in the head region altogether. And how does the skull connect to the rest of the skeleton?
An important detail from the NCERT is that the human skull is described as dicondylic. This means it articulates with the top of the vertebral column using 2 rounded projections called occipital condyles located on the base of the skull.
Okay, dicondylic skull. Let's move down to the structure supporting the skull, the vertebral column, or spine.
Right, the backbone. It's formed by a series of 26 individual units called vertebrae, all lined up serially. It runs dorsally along the back from the base of the skull down to the pelvis and forms the main structural support for the trunk.
And it protects something vital, too.
Yes, crucially, the vertebrae are shaped to form a hollow central neural canal that runs through the entire column, providing a protective bony passage for the delicate spinal cord.
And the first vertebra has a special job.
Yes, the very first topmost vertebra is called the atlas. It's specially modified to articulate with those 2 occipital condyles of the skull we just mentioned. This joint allows you to nod your head yes.
Okay. Now, the vertebral column isn't uniform, is it? It's divided into regions.
That's right. The NCERT breaks it down based on location. Starting from the top, just below the skull, you have the cervical region in the neck, then the thoracic region in the chest, the lumbar region in the lower back, followed by the sacral region, which is actually 1 fused bone in adults formed from 5 embryonic vertebrae, and finally the coccygeal region, or tailbone, also 1 fused bone in adults from 4 embryonic vertebrae.
And the numbers in each region.
7 cervical vertebrae, C1, C7, 12 thoracic vertebrae, T1, T12, 5 lumbar vertebrae, L1, L5, 1 sacrum, 1 coccyx.
And the NCERT points out something interesting about the cervical number.
Yes, it highlights a fascinating point of mammalian anatomy. Almost all mammals, regardless of neck length, think a mouse versus a giraffe, have exactly 7 cervical vertebrae. It's remarkably conserved.
Wow. So main functions: protect spinal cord, support the head, and provide attachment points.
Exactly. Attachment points for the ribs, which form the next part of the axial skeleton, and also for the large muscles of the back.
Right, let's talk about the rib cage. It involved the ribs, the sternum, and part of the vertebral column.
Correct. You have 12 pairs of ribs. Dorsally, each rib connects to one of the 12 thoracic vertebrae. Ventrally, at the front, most ribs connect to the sternum, which is that flat bone running down the midline of the chest.
And the ribs themselves have a specific feature at the back.
Yes, the NCERT describes them as bicephalic. This means each rib typically has 2 articulation surfaces, or heads, on its dorsal end where it connects with the thoracic vertebra, providing a more stable joint.
Okay. Now, how they connect at the front to the sternum determines how they're classified, right? True, false, floating.
Exactly. It's all about the ventral connection. The first 7 pairs, ribs 1 through 7, are called true ribs. They attach directly to the sternum via strips of hyaline cartilage, specifically costal cartilage.
Direct connection, true ribs. What about the next few?
Ribs 8, 9, and 10 are called false ribs, or more specifically, vertebrochondral ribs. They don't connect directly to the sternum. Instead, their cartilage connects to the cartilage of the 7th rib, which then connects to the sternum, so an indirect connection.
Indirect connection, false ribs, and the last 2 pairs.
Pairs 11 and 12 are the floating ribs. They are the shortest, and they are only attached dorsally to the vertebrae. They have no connection ventrally at all. They don't reach the sternum or any other cartilage. They just float.
Hence the name. Okay, so that covers the 80 bones of the axial skeleton. Now for the limbs and the structures that attach them, the appendicular skeleton, how many bones here?
This comprises the bones of the limbs, arms, and legs, and the pectoral, shoulder, and pelvic hip girdles. There are 126 bones in total here. Each limb, arm, or leg contains 30 bones.
30 bones per limb. Let's break down the forelimb, arm, and hand first.
Starting from the shoulder joint, you have the humerus in the upper arm. Then in the forearm, you have 2 bones, the radius and the ulna.
Humerus, radius, ulna. Then the wrist.
The wrist is formed by 8 small bones arranged in 2 rows, called the carpals. Then comes the palm of the hand, which is supported by 5 longish bones called the metacarpals. And finally, the fingers, digits, are made up of 14 small bones called the phalanges, 2 in the thumb, and 3 in each of the other 4 fingers.
Okay, 1 plus 2 plus 8 plus 5 plus 14 equals 30 bones. Makes sense. Now for the hind limb, leg, and foot. Similar pattern.
Very similar basic pattern. Starting at the hip joint, you have the massive femur or thigh bone, the longest and strongest bone in the body. Below the knee and the lower leg, you again have 2 bones, the larger tibia, shin bone, and the thinner fibula.
Femur, tibia, fibula. What about the ankle and foot?
The ankle is formed by 7 bones called the tarsals note. 7 here, not 8 like the carpals. Then you have 5 metatarsals forming the sole or arch of the foot. And again, 14 phalanges making up the toes, 2 in the big toe, 3 in each of the others.
But wait, 1 plus 2 plus 7 plus 5 plus 14 equals 29? We're missing 1 bone compared to the arm.
Good catch. The 30th bone in the leg is the patella, or kneecap. It's a small, cup-shaped bone situated on the ventral side of the knee joint, embedded in the tendon of the thigh muscle.
The patella makes 30. Okay. Now, how do these limbs connect to the axial skeleton? We need the girdles. Let's start with the pectoral girdle, the shoulder.
The pectoral girdle helps articulate the upper limb with the axial skeleton. Each pectoral girdle, you have 2, 1 for each arm, consists of 2 bones, the clavicle and the scapula.
Clavicle is the collarbone, right?
Yes, the familiar collarbone at the front. The scapula is the large triangular flat bone located on the dorsal back side of the thorax, commonly known as the shoulder blade. The NCERT notes it typically lies between the 2nd and 7th ribs.
And the scapula has some distinct features.
It does. It has a prominent sharp ridge on its posterior surface called the spine. This spine flattens out laterally into an expanded process called the acromion. The clavicle articulates with this acromion process.
So the clavicle connects the scapula to the sternum, sort of bracing the shoulder.
Exactly. And where does the arm bone itself attach? Just below the acromion, there's a shallow depression on the scapula called the glenoid cavity. This cavity articulates with the head of the humerus to form the shoulder joint, a very mobile but relatively unstable ball and socket joint.
Got it. Now for the lower limbs, The pelvic girdle or hip girdle.
The pelvic girdle connects the legs to the axial skeleton and also supports the weight of the upper body. It consists of two large coxal bones, often just called the hip bones. Each coxal bone is actually formed by the fusion of three separate bones during development.
3 bones fused into one. Which ones?
They are the ilium, the large flaring upper part, the ischium, the lower posterior part you sit on, and the pubis, the lower anterior part.
And where the thigh bone connects.
At the point where these 3 bones fuse together on the lateral side of the coxal bone, there's a deep socket called the acetabulum. This is where the head of the femur articulates, forming the hip joint, another ball and socket joint, but much deeper and more stable than the shoulder.
Okay. And the 2 coxal bones connect to each other.
Yes, the 2 coxal bones meet ventrally at the front at a joint called the pubic symphysis. This joint contains fibrous cartilage, allowing for very slight movement, which is important, for example, during childbirth. Dorsally, each coxal bone articulates strongly with the sacrum part of the vertebral column. So we've got the muscles providing force, the skeleton providing levers, but to actually allow movement at specific points, we need joints. As the NCERT states, joints are the essential points of contact between bones, or sometimes between bones and cartilages.
They act as the pivot points, the fulcrums.
Exactly. The force generated by muscles is transmitted across these joints to produce movement. Locomotion, and indeed most types of movement, would be impossible without them. The type of joint determines the type and degree of movement possible.
And the NCERT classifies joints into 3 major structural forms, based mainly on how much movement they allow.
That's the primary way, yes. We start with the joints that allow the least movement, fibrous joints.
Fibrous. Sounds like they're held together tightly.
They are. These joints permit absolutely no movement. The bones are held together very firmly by dense, fibrous connective tissue. The classic example given is the sutures between the flat bones of the skull. These bones essentially fuse end to end, forming the rigid, protective cranium. Stability is the priority here, not movement.
No movement at all. Okay, what's the next category allowing a little bit of give?
Those are the cartilaginous joints. As the name suggests, the bones involved are joined together by cartilage, either hyaline or fiber cartilage. These joints permit only limited movements.
Limited movement. Where do we find these?
The key example mentioned in the NCERT is the joints between the bodies of adjacent vertebrae in the vertebral column. These intervertebral discs, made of fibrocartilage, allow for slight compression, bending, and twisting of the spine, providing flexibility and shock absorption, but preventing excessive motion.
Makes sense. So no movement, limited movement. That brings us to the joints that allow significant motion, the ones really crucial for locomotion.
Right, these are the synovial joints. They are characterized by allowing considerable movement. Their defining feature is the presence of a fluid-filled space called the synovial cavity between the smooth, cartilage-covered, articulating surfaces of the bones involved.
A fluid-filled cavity. What's the fluid?
It's synovial fluid secreted by the synovial membrane which lines the joint capsule, except over the articular cartilage. This fluid acts as a lubricant, reducing friction between the bones during movement, and also helps nourish the articular cartilage.
So this structure allows for much freer movement, but maybe less stability.
Generally, yes. There's often a trade-off between mobility and stability. Synovial joints are typically reinforced by ligaments, which are strong bands of connective tissue that connect bone to bone, helping to stabilize the joint and limit excessive or unwanted movements.
Okay. The NCERT must give examples of different types of synovial joints based on the kind of movement they allow.
It does. It highlights several important types, demonstrating the versatility of this joint structure. For instance, the ball and socket joint. Here, the rounded head of one bone fits into a cup-like socket of another. This allows movement in multiple planes, including rotation. Think of the shoulder joint, humerus and glenoid cavity, and the hip joint, femur and acetabulum.
Maximum mobility. What about simpler movements?
Then you have the hinge joint. This works like the hinge on a door, allowing movement primarily in only one plane flexion and extension. The elbow joint and the knee joint are classic examples.
Okay. Any others?
Yes, the pivot joint, where a rounded or pointed part of one bone fits into a ring formed by another bone and ligaments. This allows only rotation around a central axis. The joint between the first 2 cervical vertebrae, the atlas and axis, which allows you to shake your head and no, is a prime example.
Rotation. What about sliding?
That's the gliding joint, also called a plane joint. Here, the articulating surfaces are usually flat or slightly curved, allowing for sliding or back and forth movements, but limited rotation. Examples are found between the carpal bones in the wrist and the tarsal bones in the ankle.
And there's one more specialized one mentioned for the thumb.
Ah, yes, the saddle joint. This is found at the base of the thumb, specifically between the trapezium, a carpal bone, and the first metacarpal bone. Each articular surface has both concave and convex areas, fitting together like a rider in a saddle. This unique structure allows the thumb to move in two planes and is crucial for the ability to oppose the thumb to the other fingers, essential for grasping.
Fascinating variety. Okay, to wrap up our deep dive, we should briefly touch upon what happens when these intricate muscular and skeletal systems malfunction. The NCERT lists several common disorders.
Right. It's important to understand the implications when things go wrong. We can broadly group them. First, disorders primarily affecting the muscles or the neuromuscular junction. Myasthenia gravis is mentioned. This is a serious autoimmune condition where the body's own immune system attacks receptors at the neuromuscular junction, specifically for acetylcholine.
So the signal transmission fails.
Exactly. This leads to characteristic symptoms like fatigue, progressive weakening, and even paralysis of the skeletal muscles. Then there's muscular dystrophy. This isn't autoimmune. It refers to a group of genetic disorders that cause progressive degeneration and weakening of skeletal muscles over time.
And what about uncontrolled contractions?
That condition is called tetany. It's characterized by rapid, involuntary spasms or wild contractions of muscles. The NCERT notes this is often caused by low levels of calcium ions, Ca2+, in the body fluids. Calcium is essential for regulating muscle excitability, so when it's too low, muscles can become hyperexcitable and go into spasm.
Okay, now moving to conditions affecting the joints and bones themselves.
A very common one is arthritis. This is really a general term meaning inflammation of one or more joints, causing pain, swelling, and stiffness. There are many different types of arthritis. Another major issue, particularly relevant with aging, is osteoporosis.
Bones becoming weaker.
Yes, it's characterized by a significant decrease in bone mass and density, making the bones porous and brittle. This leads to a greatly increased risk of fractures, even from minor falls or stresses. The NCERT specifically points out that decreased levels of estrogen, often occurring after menopause in women, is a common contributing factor.
A major age-related concern. And the last one listed, gout.
Gout. This is another type of joint inflammation, but it has a specific metabolic cause. It results from the accumulation of uric acid crystals within the synovial joint cavity. Uric acid is a breakdown product of purines, found in DNA in certain foods. When levels get too high, sharp crystals can form in the joints, triggering intense pain and inflammation, often affecting the big toe initially. #tag outro.
Well, we've certainly covered a lot of ground today. We started with the almost imperceptible streaming of protoplasm in a single cell and journeyed all the way through the intricate molecular ballet of the sliding filament theory, explored the remarkable architecture of our 206 bone framework, and examined the variety of joints that allow us to move, from rigid sutures to highly mobile synovial joints. It really is staggering how much complexity underlies something as seemingly simple as taking a step.
It truly gives you an appreciation for the evolutionary refinement of this entire system. Specialized tissues generating force, hardened structures providing leverage, all synchronized with incredible precision. It's a masterpiece of biological engineering.
Absolutely. Now, as we wrap up this deep dive, we want to leave you, our listeners, with a final thought to ponder something building on the details we've pulled from the NCERT material.
Okay.
We learned that the vertebral column is segmented into distinct regions, like cervical, thoracic, lumbar, and so on. But we also heard that specific point from the NCERT about the number of cervical vertebrae, the neck bones. It's almost universally fixed at 7 across nearly all mammals. Think about that, a tiny shrew, a human, a massive giraffe with its incredibly long neck, all having just 7 cervical vertebrae.
It's quite counterintuitive when you think about the giraffe's neck length.
Exactly. So the question for you to mull over is this. What unique evolutionary pressures, or perhaps developmental constraints, might have been so incredibly strong as to conserve that precise number 7 cervical vertebrae across such vast differences in body size and neck length over millions and millions of years of mammalian evolution?
It certainly suggests there's a deep underlying blueprint at work, doesn't it? Something fundamental in the developmental plan. A fascinating question to consider.
Indeed. Thank you for joining with us today.
See you next time as we continue the deep dive into the NCERT material.